So in this video, I want to talk to you about the first part of the central dogma of biology, where DNA is converted to RNA. And of course, we call this process transcription. Now, transcription is broken up into three sub-stages: initiation, elongation, and finally, termination. Okay. So, these three stages feed into each other. And let's talk about what's required for each of these to happen. So, for initiation, on my DNA, there is going to be a distinct region that's going to signify the start of a gene. And this region is classically referred to as a promoter. So I'll draw it here in green. So here's my DNA, and this of course is called the promoter. This is the site where we're going to initiate transcription. So there's the promoter, and let's go ahead and label our 5' and 3' ends. So here's 5' at top, 3' at the bottom, which means over here this is going to be 5' and 3'. Okay, so the first thing I always need is a promoter. So that's the first thing that I'll need for initiation. Now, within that promoter, if we blow it up just a little bit, in eukaryotes, there's going to be a distinct region called a Tata Box. That ties into part of the promoter. That's going to be a very critical part for our transcription. Okay, so here again is my 5' end, 3' end, and it's called the Tata Box because we have the alternate sequence TA-TA. Okay, so promoter, and within that promoter, there's the Tata Box. Now, the Tata Box is critical for us because it's ultimately going to result in the recruitment of a class of proteins that are called transcription factors. So those transcription factors will bind to the Tata Box and to various regions of the promoter. So these three things that we have to have before we can even initiate transcription are the promoter. Within the promoter, in eukaryotes, we need the Tata Box. And then we're going to also need these transcription factors. Okay. Now, once we have all three of these things, we're going to be able to initiate transcription. And this is going to be achieved. So I'm going to redraw the promoter and the Tata Box like this, just so you understand. Prime. Here is my promoter. And then here are my transcription factors attached. And when I have all of these things together, that is going to ultimately recruit RNA polymerase 2. So this is RNA polymerase 2. So to get RNA polymerase 2, I have to have these three things. And all of this together, the transcription factors, the Tata Box, the promoter, and the RNA polymerase 2, all of that together is classified as the transcription initiation complex. And so once I have all of this, that RNA polymerase is going to start synthesizing RNA off of one of the two strands. Okay, and again, just like DNA polymerase, it's going to synthesize it from the 5' end to the 3' end. Now, the strand of DNA that is used to make this piece of RNA is commonly referred to as the template strand. Okay, so here my polymerase, it begins to move in this direction, and it's synthesizing that new piece of RNA, which I've drawn in red. So, as it goes along, ultimately what the polymerase does is it systematically unwinds the double helix as it moves. So unlike DNA replication where we need a helicase, once RNA polymerase initiates transcription, it's going to just open up the double helix about 10 to 20 nucleotides at a time as it continues to synthesize RNA. Go ahead and just draw my ends here. And so it's going to continue to move down this sequence, and as it does, we're going to notice that the piece of mRNA, or the primary transcript that is going to get longer and longer and longer. So here, my RNA polymerase has moved farther down, and again, my piece of mRNA, or my primary transcript, is now even longer than it was before. And notice, as it moves, the parts of the DNA that it is no longer bound to zip back up. So unlike helicase, which just fully unwinds the double helix, RNA polymerase only unzips the double helix at the points where it currently is, and then when it moves over them, they zip right back up. So you can kind of think of RNA polymerase almost like a broken zipper. When you move it, it doesn't wind up unzipping the whole zipper, it only unzips it wherever it currently is. Okay, so initiation, elongation, and now let's talk termination. So for termination of transcription, it's actually going to occur in one of two different ways, depending upon if we're in prokaryotes or eukaryotes. So in prokaryotes, we're going to have our piece of DNA, and on this DNA, we're going to have a distinct region called the terminator. So this terminator sequence exists, and when the polymerase reaches that terminator sequence, so here's our polymerase still chugging along and synthesizing mRNA, but when it reaches that terminator sequence, the polymerase falls off. And when it falls off, it releases that piece of RNA that it had generated. So in prokaryotes, all that's required is for the polymerase to reach a terminator sequence, and once it does, it falls off of the DNA. In eukaryotes, however, to terminate this, okay, there is no terminator sequence, actually. So instead, what I wanted to draw was this. So there is no terminator sequence. Instead, there's what we call a polyadenylation signal. So here, it's going to be a poly-A signal. So the polymerase is going to pass over that poly-A signal, but when it does, it doesn't fall off. Instead, what happens is after it passes the poly-A signal, it's going to move 10 to about 35 nucleotides down, and then it's going to fall off. And so the mRNA, or the primary transcript that's made, will actually have that polyadenylation signal in it. And that's going to serve a really important purpose when we get to RNA processing. So the big difference here, prokaryotes, they hit a terminator sequence and the polymerase falls off. With eukaryotes, the polymerase passes over that poly-A signal, and then 10 to 35 nucleotides later, it falls off, and that poly-A signal winds up in the mRNA molecule. So this is transcription, initiation, elongation, and termination.